not mistaken.
Yeah, let's start.
Okay, great. Greetings, everyone. Thank you for joining us today. And it's with great pleasure to introduce the host and moderator, Dr. Alexander Satz, who is Senior Director of Dell Strategy and Operations at WuXi AppTec. Most of you may know Alex already through your research. Alex's contribution to the field included developing the first industrial scale Dell platform at Précis Pharmaceuticals, and then moved on to build the Dell platform at Roche in Basel. Alex has over 30 peer-reviewed publications, and the most recent of which is quite interesting. It describes a method to triage hits from Dell using ASMS. Over to you, Alex.
So thank you, Suzanne, for the introduction. And I'd also like to thank the entire Target 2035 team for hosting us here. I would also like to thank all of you who have signed in and who are joining us now. We are 4 of us. 3 of us are going to be giving short presentations followed by a Q&A. So that's our job, but we have a very important job for you as well. And that is during these presentations, if you have any questions or something that you don't understand, please go to your Q&A box at the bottom and type that question in. We'll have a 5-minute Q&A after every single presentation. And then at the end of all 3 presentations, we will have a panel discussion where we will try to answer big questions such as, why is Dell and why is Dell screening a perfect way to find hits for those of you working on the Target 2035 project. Okay, so, and I also want to point out that this presentation will be recorded. So that's something that all of you should know as well. Okay, so with that said, we will jump right into the science. I would like to introduce Dr. Xiaoyu Lee. He is a professor at the University of Hong Kong, and he received his PhD from the University of Chicago, and he did his postdoc at Harvard. And between his academic training and his current academic position, he also worked at several different biotechs. Xiaoyu works on very creative versions or areas of Dell and Dell screening. For instance, he has recently published a paper where he found a way to screen and find hits against targets being expressed on the surfaces of live cells. So with that, I will hand this over to Dr. Lee.
Thank you very much for the introduction. So let me share my slides. Okay, so thank you very much. Thank you very much for attending this webinar. So first, I have to apologize that the title of my presentation is a little bit different from on the poster. So today, I would like to spend some time to talk about some of the new methods we have developed for the synthesis and selection of DNA-encoded chemical library in the past few years. And since I am the first speaker today, so I guess I could spend a few minutes to give you a very brief introduction of Dell. So we all know that traditionally, in a traditional chemical library, the compounds are spatially encoded. So a robotic system is going to screen the compounds one by one, but with Dell, each compound is conjugated with the DNA tag, which encodes the chemical structure information for that particular compound. So this kind of DNA encoding, it brings in 2 benefits. First, it enables the processing of the entire library in a single solution. There is no need for spatial separation. Second benefit is the DNA. It's compatible, it's a biomolecule, it's compatible with the PCR modification and the sequencing technologies. So you could synthesize and select the library at a very small scale, so atomal or femtomorph compound, even less. So in a very small epitope tube, and it can hold a Dell with hundreds of millions to even billions of compounds. So the idea of Dell was proposed by Brenner and Lerner more than 30 years ago, almost 30 years ago. And in the past few decades, there has been some very significant developments of Dell on many fronts. So actually now the DNA can be used to encode not only just chemical structures, it can be used to encode the outcome of chemical reactions, functions of catalyst, enzyme activities, and you can also use DNA to encode the configurations of ligand binding against biological targets. But I guess the most famous feature of DEL is large size. Now you can prepare DEL with hundreds of billions to even trillions of compounds. And also with the recent very rapid development of DEL-compatible chemistries, and you can build a DEL with all kinds of structures now, way beyond the peptides, like drug-like structures, macrocycles, natural product scaffold-based DELs, or even DNA-encoded polymer libraries. And there are several different kinds of formats of DELs. For example, the dual pharmacophore DELs. I think Dr. York Schuyerman going to talk about this today, later. And I will talk about, briefly talk about our recent work on the dynamic DELs. And there are also antibody mimicking DELs. And very recently, the so-called one bead, one compound DELs, mostly developed by the PAGO and by the research group of PAGO and Kodadek. So the DEL have been widely adopted by most of the pharmaceutical companies in drug discovery. So that's background. So as a small academic research group, we've been mostly interested in methodology development. On the one hand, we try to develop a new encoding method to synthesize DELs. On the other hand, we try to develop a new selection method to select DELs against all kinds of targets. Then we apply these methods in different applications. So first of all, I would like to talk about our recent work on the encoded dynamic library. So the dynamic combinatorial library, DCL, not DEL, is a classic type of combinatorial chemical library. It has been around for a few decades and pretty useful in ligand discovery, material chemistry, and system chemistry, etc. For DCL, you do not have to make all the compounds in the library. You just throw all the building blocks together in a single solution, and they undergo reversible chemical reactions. And all the compounds in the library, they are synthesized automatically in situ in a single solution. Then you add the target in the solution, which can facilitate the formation of the high affinity ligand at the expense of the non-binders. So everything is under small dynamic control. So if you are able to compare the equilibrium before and after the target addition, then you will be able to identify the hit compound. So that's DCL. So as I mentioned, DCL has been around for quite a few decades, but there is a major and also long-lasting limitation, small library size. If you look up the literature, most DCL, they have less than 100 compounds. Why? It's just because, as mentioned in this very recent review on DCL, it's because the current analytical methods are unable to identify the best binders from large and complex dynamic libraries. So it's desirable to develop high throughput screening DCL methodologies. So that's exactly what we have been doing in the past few years. Because the DCL and DEL, they both process all the compounds in a single solution, but with the DNA encoding, it can contain a lot of many compounds, a large number of compounds in the library. So if we're able to combine DEL with the DCL, then we get DNA encoded dynamic library DEL. Probably can address the library size limitation for the classic DCL. So here's our, if you may call it, the first generation of this method. So the idea is very simple. We took a set of DNA encoded fragments. Then we hybridize with anchor DNA. We call it anchor DNA because it contains the known binder for the target you want to select. And this anchor DNA is very short, so only 6 bases. So it forms a dynamic, very short DNA duplex. It's not very stable, so it's on and off, on and off. It's a dynamic duplex formation. Then the target addition going to shift the equilibrium to increase the formation of the high affinity duplex. Then photo cross-linker and the distal end of the anchor DNA and the UV irradiation, it can capture the other DNA strand. So it locks the equilibrium. So the cross-linker duplex could be isolated and then subject to sequencing to deconvolute the fragment selected. So this idea is very simple if you look at this scheme. And it's of course, it's limited to single fragment display. It's not a true combinatorial library, and it can only used for affinity improvement of known ligands. So in 2018, we developed another, you may call it second generation. So now we can use 2 sets of DNA encoded fragments to form a real combinatorial combination of the fragments in the 2 sets of the DNA encoded small molecules. Then the addition of the target are going to shift the formation, are going to promote the formation of high affinity duplex, very similar to the last case. And we used a very special base called the P-stereobenzyl base developed by a group, I think, in Nagoya University of Japan. So it's it can, if you put this special base in the DNA duplex, they can cross-link to each other to lock the equilibrium. And the cross-linked DNA duplex could be, of course, isolated and subjected to heat decoding to identify the fragment pairs. But the selection will only give you the identity of the fragment pairs. There is one additional step you have to do, which is fragment linking. You need to find the proper linker to connect them together, then test for the binding affinity and the biological activity. So this fragment linking step is not trivial. It's pretty difficult, actually. It's empirical, it's laborious, and it involves a lot of trial and error. So to solve this problem, more recently, we developed, if you may call it 3rd generation of DEDL. So we took a regular DNA encode library, but this library has a amino group. This amino group, through this amino group, it can form a imine group, form an imine linkage with the anchor molecule. So again, this anchor molecule is a known ligand for the target you want to select. So you have this dynamic assembly of this library. Very similarly, the target addition going to increase the formation of the high affinity imines, and then through the sodium cyanide borohydride, you can reduce imines to amines, so you could make the lock. Then you can do the heat deconvolution. So here you do not have to do the fragment linking. You can obtain the full ligand structure directly from the selection. So up to now, you can see we have developed 3 different versions of DEDLs. The first version is very simple, very basic. It requires fragment linking. It requires a known ligand. The second version is better, and it can be used for novel ligand discovery. You do not need a known ligand, but it does require fragment linking after selection. The 3rd generation, we eliminated the fragment linking step, but unfortunately, we had to take 1 step back and again use a known ligand in the selection. So currently, we're working on the 4th version. The project is almost done, and hopefully, we'll be able to report the result early next year. In this version, we are trying to solve 2 problems. First, no fragment linking. Secondly, it does not require known ligand. So the basic idea is to put the linker directly in the library and also use DNA to encode the linker so that the target will select not only the linker part, but also the fragments. So hopefully we'll be able to report this result early next year. Okay, that's the first part of my talk. Now I want to switch gear a little bit to talk about our recent work on cell selection methodology development. So recently we have been very interested in selecting cell against membrane proteins on live cells. So we all know that membrane proteins are super important, especially in drug discovery, like GPCRs, ion channels, they are major drug targets. So altogether, they actually account for more than 60% of the target of all approved small molecule drugs. But working with membrane proteins is very difficult because they are very large and hydrophobic. So they rely on this hydrophobic lipid bilayer environment of cell membrane to maintain their structures and functions. So even if you can purify them, they may often lose important biological features such as post PTMs and cofactor binding and the complex formation, etc. So ideally, if you want to do down selection against membrane proteins, the best way, the ideal way to do it like this. Okay, take a live cell, so in its native state, and incubate the DEL, and you wash away the non-binders. Then you can use strong conditions to elute the binders. So with this scheme, it looks very simple, very straightforward, but actually there are two major problems. So one is target specificity, because there are a lot of stuff on the cell surface, not just your target. So the first issue is how to achieve target specificity. The second issue is the target concentration of the effective molarity. So in the DEL selection, typically we need a relatively high concentration of proteins to drive the binding equilibrium. For example, in the high micromolars, sometimes even low millimolar. But the typical concentration of the most membrane proteins, they are in the low nanomolar range. So you just do not have enough target receptors on the cell surface to drive the equilibrium. So in the past few years, the different groups have used different have used the strategy of target protein overexpression to address these issues. Because overexpressing the target protein, on the one hand, you increase the target molarity, the effective molarity of the protein on the cell surface. On the other hand, you can always compare with the negative control cell where you do not overexpress the protein target and to identify the target specific ligands. For example, in 2015, the GSK reported the first cell-based DEL selection where they overexpressed this NK3 target on the cell surface, and they selected a bunch of DELs against this protein. This is on the left cell, and compared with the negative control cells. And in the 2019, the Krusmark group at Purdue University, similarly, they overexpressed this delta opioid receptor on cell surface and use the covalent cross-linking strategy and conducted DEL selection. And more recently, the Schuyerman and Nero group, they systematically and comprehensively optimized the cell-based DEL selections using the carbonic anhydrase 9 as the model system. And also, in addition, it also was mentioning that the Krusmark strategy, in Krusmark's report, they also realized that the intracellular DEL selection by using a cell penetrating peptide to deliver the DEL into inside the cells. And finally, also very recently, the company of Vibergen, they also performed the intracellular DEL selection by directly injecting the library into cells through microinjection. But in all of these cases, the protein target, it was overexpressed to improve the effective molarity and also to compare with negative controls. And we were thinking, is there any way to perform DEL selection on live cell against membrane protein without overexpression or any other genetic manipulation? So because some of membrane proteins, they are not very happy, they are not compatible with overexpression, and there are some cell types you can just you just cannot overexpress your target. So how to do this? We were thinking, is there any way to add a DNA tag on the target? So it's like adding a flag on the cell surface. The DNA tag will guide the hybridization of DEL close to the targets. So by doing this, first, we're going to have target specificity. So the DEL will hybridize based on the DNA sequence of DNA tag on your target. Secondly, the hybridization is going to place all the library compounds very close to the protein target, so you increase the effective molarity of the target for all the compounds. So it put all the compounds very close to the protein for the protein to sample the binders. But the question is how to achieve this target specific labeling of the DNA tag on the cell surface. So we have decided to use a DNA program affinity labeling called DPAL we have developed in the past few years. This method is a dual probe system with two DNA-based probes. One is called binding probe. It contains a known ligand for the target. So after the binding probe engages the target, we use a capture probe to hybridize with binding probe, and through DNA template photo cross-linking, and we can label the proteins with this tag. So before we talk about how we're going to use the for DEL selection, I just want to show a few examples how we apply this method in different applications. Actually, a while ago, we already used this method, dpol, to select DELs against the non-immobilized proteins in the solution in cell lysate. We hybridize this so-called pcDNA, photo cross-linking DNA, close to at the prime binding site of DELs. Then we incubate it with the protein. Then the UV irradiation will cross-link the ligand, the binders with the protein. Then the addition of exonuclein 1 will destroy the DNA tag of the non-binders, and the DNA binder of the DNA tag of the binder will be protected and can be directly amplified and identified. So that's one application. Another application is on the protein profiling. Recently, we have used this dpol to achieve the profiling of HDAC complexes. So HDAC, it's called histone deacetylase. It's a major class of deacetylation enzymes. They often exist in this very large protein complexes, and the composition or structure of the protein complex is very important for the HDAC biological functions. So we use a known ligand connected with a long DNA, very long DNA. Then this ligand can engage HDAC. Then because the DNA is long, so we can hybridize not one, but multiple capture probes on the same binding probe. The idea is that we can place the photocross linker at different positions within the complex. So sometimes it may capture the HDAC, sometimes it may capture the HDAC binding proteins, sometimes it may capture a distal protein in the complex that is pretty far away from the HDAC, but it still binds to HDAC indirectly. So this type of protein is very difficult to be labeled, be captured by small molecule-based probes. So this is one of the benefits of using dpol. And we can also use dpol for target identification. So a few years ago, we identified the additional target for elicited, which is supposedly to be a very selective aurora kinase A inhibitor. But we have identified it has at least 6 additional targets. And we have also validated 2 of the targets, P38 and the laminin receptors. They are involved in the anti-cancer activities of the elicited. And recently, our group is also working on the target identification of this very interesting molecule called dihydromortopyramine C. This molecule has very simple, interesting structure. It has this hydrophobic head with a polyamine tail. It's a natural alkaloid isolated from C sponge quite a while ago. Very simple structure, but very good anti-cancer activities with relatively low cytotoxicity. So target mechanism remain unknown. And we have used a dpol and identified its target. Because this work is not published yet, which is called target A. Very interesting target A is existing, it's in the mammalian cell, but it's very poorly is very poorly characterized, and people knew very little about its biological functions and biological roles. And through this work, we might have discovered a new regulator of cell migration. So this is another application of dpol. Okay, now let's get back to the DEL selection. So as I mentioned, we have used DELs. We have used dpol. We use a known ligand to guide the initial labeling of the target of interest. Then after the we first use this binding probe to engage the target, then use capture probe to do the labeling. After labeling, we use total displacement, another DNA probe to remove the original binding probe from the cell surface. Now the binding site on the target is reopened, so it's available for DEL selection. And then we can simply hybridize the DEL and to this DNA tag. And but you can see all the compounds, they have the same green, this DNA, this sequence complementary to the target. That means all the compounds in the library will be able to hybridize to the DNA tag. Now, how to separate the binders and non-binders? We decided to use this the concept that we developed in we used in the dynamic library. We designed this the green part very short, only 6 pairs. So for non-binders, they form unstable dynamic duplex. They could be easily washed off the cell surface, but while for the binders, this duplex has the increased stability. It remains on the cell surface until you use stronger condition to elute the binder of the cell surface and to the target to the hit identification. Okay, here I just want to quickly show you one example. So we use the 30 million member cell and select against the folate receptor. So and the selection result are shown on the right side. You can see with the DNA tagged cell, we have identified a few folate receptor ligand with the binding affinity ranging between nanomolar to micromolar. But in comparison with unlabeled cells, we could only identify something else. They are not folate receptor binder. They might bind to something else on the cell surface. Okay, so this method, it works, but it has one major limitation. You do need a known ligand at the beginning to guide the labeling. So this is the limitation because this kind of ligand, high affinity, high spectral ligand may not be available for your target. And then we spend quite a lot of time trying to use antibodies because antibodies, they are relatively more available for membrane proteins. So I'm going to skip you all the technical details here, but in the end, we can use antibody to guide the labeling. So here I just show you one example. Anti-EGFR, we use anti-EGFR antibody to guide the labeling. Then we selected the DNA tagged EGFR against the same DEL, and we found some EGFR inhibitor, EGFR ligand, and they have the low micromolar affinity. Okay, finally, I just want to quickly show you that recently we have extended the application of this DNA labeling to the selections of non-DELs, so non-DNA conjugate small molecules. So we reason that after the labeling, okay, you have a DNA tag on the target. You could hybridize the FP fluorescent probe, which contains this known ligand and the fluorophore on the other end. So it will increase the cell fluorescence. But in the presence of small molecule competitors, it will displace the FP, the fluorescent probe, from the cell surface. It will reduce the cell fluorescence. So for example, as I showed here, so if we use FA, folic acid, a known ligand for the folate receptor, it reduced the cell fluorescence from around 19% to about 20%. But other hydrocycles, they are not folate receptor ligand, so they have very little effect on the cell fluorescence. And we have used this method to select a collection of 200 natural products and bioactives, and this method against the folate receptor. And we have found there are non-compounds, they reduced the cell fluorescence more than 20%, and the 3 of them are shown on the right side. The first one is not a surprise. It's a levoglucovorin. So this structure is very similar to folic acid. So it is a folate receptor ligand. But the other two are very interesting. This one has a very complex structure. This one has a very simple structure. But they are using fluorescent polarization as an orthogonal way to validate their binding. So they are folate receptor binders. You can just imagine if you want to connect these two with the DNA, it's not that easy. So, and another benefit from this method, you can just titrate the concentration of the competitors. So you'll be able to obtain the IC50 directly from your selection. So this is another application of this DNA tagging strategy. I think I'm already, I ran out of time already, so I guess I stop here. And finally, I want to thank my students and my collaborators and my funding sources for their support. Okay, finally, thank you very much for your attention.
Great, thank you. Yeah, very interesting talk covering many different topics. We don't have a lot of time, but there is one question that I would like to ask. And that is, when you do these screens, you have this 6 base pair region, which stabilizes the complex, I suppose, when it's bound to the target of interest. And could you tell us a little bit about how you played around with how long a region that was going to be, and to what extent different cell lines and different buffers might have an effect on how effective a 6 base pair region is?
We did play around with the length of the tag, but not so much with different cell lines and buffer conditions. So with the length, and here's a couple things that we should consider. First, it cannot be too short. Otherwise, you don't have specificity. So I consider 5 is like the minimum, but it's already on the borderline. So we start from 6, I think. Then we test 6, 7, 8, 9, I think up to 10. And we also reported that we also did some calculation. It's in the paper. So when you go to 10, it becomes so stable, quite stable. It becomes a little bit more difficult to distinguish the bundled from non-binders. But we did test the 6, 7, I think the 8 probably in the paper. We found 6, 7, they are the, you may call it sweet spot. Okay, and there is another semi-quantitative way to guide you as a guide to tell you how what the length should be and what kind of sequence. There is a calculator, so tells you okay how much energy you can gain by this DNA tag. Then you can the energy can be converted into KD. That's the KD additional binding affinity for all the library compounds. So now then you can have an idea, okay, whether in this condition I want the nanomolar binder, okay, probably it's going to be 6. The other condition I want the micromolar binder, so probably this one or 6. I want the micromolar binder, probably I want 6. Nanomolar binder maybe go a little bit longer. So.
Okay.
Yeah.
I think I've got it. And of course, we always want the nanomolar binders, basically, which doesn't mean we will find them, but that is what we would like. Okay. So Xiaoyu, I know that you can't stay for the panel session. So again, thank you for being able to join us and give your presentation. And again, I would remind all the audience members, if you have any questions, please feel free to type them into the Q&A box so that this is a more interactive back and forth. It being a virtual seminar, the best we can do is have a Q&A. Okay, so up next, we have Dr. York Schuyerman. York is currently a principal investigator at ETH Zurich, where he oversees a fairly sizable group doing DEL research. Of course, he received his PhD at ETH Zurich as well, and he's actually been working in the DNA encoded field now since about 2002, so almost 20 years. I think it's safe to say that York was one of the original founders of DEL, at least in the academic sense. So he's been working in this field from the very, very beginnings. Okay, and the title of his talk is DNA Encoded Libraries, Present and Future Developments. So York, I will hand it over to you.
Thank you, Alex. Can you hear me? Thanks, Alex, for this kind introduction. And thanks for inviting me to give here my contribution to this webinar. And thanks. Yeah, I hope I can make some things clearer for you. So I'm going to talk about DNA encoded chemical libraries, more or less like I see the present and the future development. You have seen already now from Xiaoyu quite impressive, innovative research on dynamic libraries. Maybe I have to switch here back on these dual display libraries, as you will see also in my presentation. So Xiaoyu already did my introduction into the field, so I don't have to overdo it. Just very briefly, what are DNA encoded libraries? Everybody knows that, let's say, the history of synthesis of a single compound can be noted down in a respective barcode, which can then be PCR amplified because PCR is very sensitive and can be sequenced. And by that, excuse me, and by that, we can go towards this implementation. Can you see the, is it fine to maybe like that? Then you can see this implementation of DNA encoded small molecules, which essentially the field lies between, let's say, the brute force high throughput screening of individual organic compounds, which was and still is popular for finding small molecule hits. That was then later expanded towards parallel synthesis, split and mix based combinatorial approaches. Before the encoding of these compounds essentially was considered to be very important. Encoding was generally delivered by the natural display systems already, like phage display, mRNA display, and ribosome display, and you name them. And there are, now we are in this interface here with our DNA encoded chemical libraries, which in principle can span between the field between small molecules and larger polypeptide molecules. And the idea, of course, the dream was to have libraries of chemicals sized of millions to billions just in a single tube instead of having to use the big infrastructure for screening. The initial idea, as you are probably all aware, came 1992 in a theoretical paper by Brenner and Lerner, and that was actually, they foresee the synthesis, the alternate synthesis of a polypeptide chain and of the DNA chain on a bead support, and that, however, was kind of difficult to be implemented because of the not really compatible chemistries of DNA synthesis and peptide synthesis at that time. So it took some time until it was brought back to something useful. And that was essentially two approaches. To name the first one, that was DNA templated approach by David Liu from Harvard, where essentially you created a template library with individual codes, and based on these template libraries, you would then do the chemistry by just, let's say, annealing reagent oligonucleotides carrying building blocks and transferring these building blocks onto the growing chain. This is essentially a very cool technology, and it was used to create smaller macrocyclic libraries, but it was so far never used to produce, let's say, ultra large libraries. And that is kind of a limitation. There was also some other approaches, like a routing approach by Harbury in 2004. And later on, another important approach was presented with this three-way junction approach from Vipergen, a company which is still in the Dell field. So in 2004, at that time, we started essentially doing the first publications on recorded DNA encoded libraries, and we came up with a concept which is called dual display, which is the encoded self-assembling chemical library technology, where you just have two sub-libraries carrying a building block A on one DNA strand and the building block 2 on another DNA strand. Because of the DNA hybridization, you can assemble these sub-libraries combinatorially, and therefore you reach larger library sizes. And these are kind of fragment-like approaches, and we have been able to identify some, let's say, interesting compounds, like an albumin binding compound, which is essentially still now in clinical development, and which prolongs the half-life of any kind of molecule in vivo, actually. So the DNA recorded library field is essentially what is the one practiced by industry nowadays. And that means that essentially you do synthesis, and then later on you do encoding of the synthesis. And we would like to differentiate these approaches of DNA recorded libraries into single pharmacophore libraries. That means where only a single chemical molecule is displayed on one DNA strand. And this DNA strand could be either single-stranded format, or what is much more popular, at least in the industrial setting, is the double-stranded format. And there is the so-called dual display or dual pharmacophore library setup, like the ESAC setup that I've mentioned before, where essentially each of the pairing DNA strands carries one building block separately. You know better than me that these libraries are produced by split and pool-based combinatorial synthesis, so that you can do first one reaction. So you split for the first reaction, then you do the first reaction, you encode the first reaction, you pool them again, then you split for the second reaction, do the second reaction, encode the second reaction, pool again. So that is straightforward. What is important is also the selection process, and I come to that later. And Xiaoyu has already presented some very sophisticated implementations. Here is the basic one. So you just have an immobilized target. You incubate your DNA-encoded library of interest. You wash away binders which are not binding so that the ones which are still sticky to the target can be PCR amplified preferentially afterwards. And then this PCR amplicon is submitted to DNA next generation sequencing, usually Illumina-based sequencing. And then the counts are read, and the results can be presented, for example, in plots where you have, let's say, the individual building blocks on one axis, like code A and code B, and the counted sequences, or how often each sequence was counted on the Z-axis. And then you can, for example, see that, okay, some binders are preferentially enriched. So if I go now into this differentiation, which I've presented before, like single pharmacophore versus dual pharmacophore libraries, we have been active essentially in both fields. The single pharmacophore libraries, again, display kind of lead-like compounds. So they can be produced by split and pool, theoretically also by template-assisted synthesis. The problem always is that after one coupling step, the follow-up coupling steps like 2 and 3 usually are less efficient and usually cannot be anyway seen by any analytical method. So you have to believe that your library is good. So we had been active in this field as well, and I think we were the first who published a library of a split and pool based library in literature in 2008. Recently, we also have published some, let's say, more interesting thing where we looked into exploring this area on radioselectivity of our scaffold, so to speak. That means we had a scaffold which projected a building block first to one lane or to the other plane. And then on the other hand, we additionally had some radioselective attachment of the second building block, either in parameter or ortho position. The library size was 670,000 compounds, if you count them all together, which does not seem very large. But as I've mentioned before, the neat library size does not say anything about the quality of a library. So looking at the fingerprints, so the results of these libraries here, you can see, for example, no protein controls, which are quite uniform. Then you can see we have always produced selections in duplicate. So this is a target of interest, tumor-associated target. And what we see is, for example, lines of different building blocks here, A, and you can see how the, let's say, parameter and ortho position is important, giving already some kind of hints for future structure activity development. Also, this was also to show that DNA-encoded chemical libraries can be used to find ligands, for example, which then can be linked, for example, to fluorescein, and therefore being used for, let's say, interesting tumor targeting approaches like universal CAR T-cell technology, where you have, let's say, an antigen on your cells which is overexpressed, and you have a binder towards this antigen, which you can fuse, for example, to fluorescein, and together with the universal CAR T-cell, which displays an anti-fluorescein antibody, you can create let's say, an artificial synapse between the tumor cell and the killer cell, which eventually leads to the decay of the cell. So that could have some practical implementations. But what we're starting with also in 2004, as I mentioned before, is the dual display approach, where you have, let's say, two different compounds on each end of the DNA heteroduplex. If it is only one compound, it's just one building block, it's more fragment-like. And therefore, as Xiaoyu pointed out, at some stage you have to find a linker which links these two compounds. But this technology proved to be, let's say, useful, and it also creates quite kind of large libraries. For example, if you have just one building block and you have a library of 10 to the X and a sub-library of 10 to the Y, you can pair them to a library of the size 10 to the X plus Y. And if you go into a direction which we are actually going, where we combine a single pharmacophore library with two building blocks with another library of one building block, then we can end up with a very large library size, easily going into the billions. This SARC technology or the dual display technology also has been of practical use. We have found, for example, a ligand which in combination with already good ligand, acetazolamide, which is a 20 nanomolar binder to carbonic anhydrase 9. And you can see this is already located at the tumor very clearly, but it fades away because of the 20 nanomolar. So we have found an additional fragment here, which essentially improves the affinity of the whole compound towards CA9 by a factor of 40 at least. And you can see this, so it's 0.5 nanomolar, and you can see this is reflected by in vivo experiments. So the residual time on the tumor, the residence time on tumor is much larger than for the 20 nanomolar binder, and this of course might have also some therapeutic importance. And also, if you go into small molecules, you know, we were recently talking always about antibodies as specific compounds which can be raised against any target of interest. Antibodies usually never exceed 10% injected dose per gram. So if you inject them and then they locate at the tumor maximum at this 10% injected dose per gram, while small molecules can better diffuse into the tumor and can lead to rocketing amounts of material being deposited in the tumor. So that small molecule conjugates, drug conjugates, for example, have some potential for therapeutic use. And this molecule, which I've shown you before, the CA9 binder, essentially is also in clinical trials. And you can see here it is able to target well the tumor. I've mentioned also before that we are going into, let's say, kind of a 2+1 setup where we have one sublibrary with 2 building blocks, one with 1 building block. We created a larger library. And I wanted to say that this approach essentially, so what industry does, for example, is having like small single pharmacophore libraries, which usually are double-stranded, so that is essentially kind of a hurdle. And then it is selected and the PCR amplified and just submitted to sequencing. But if these libraries would be single-stranded, you could use it to very sophisticated selection approaches, as you've heard from Xiaoyu Lee, who's actually the expert in that, by pairing this library, if it is it's only possible if it's single-stranded, with, for example, a photoreactive group, which then can be linked at the target site, or a reversible covalent group like salicylaldehyde, for example, which could engage with a lysine at the binding site, or even, I don't know, let's say covalent group, for example. This can be used to increase, let's say, the potential of single pharmacophore libraries, but it can be also used to create libraries of its own by, for example, matching this single pharmacophore library with an own library here on as building block 3, or already as a known hit. So something that you already know and that you would like to make better. And we have shown recently that this in principle works well. So if we had found a hit, we could, let's say, by pairing the libraries together, we could find a structure, AB, which essentially optimized this lead. And that can be shown, for example, here, where this hit validation was done then on DNA. And you can see here the hit together with the one is, of course, better than the hit alone. So you find always the compounds from a library which improve your hit, and then you can go from that as a new starting point. And that has been shown on different targets. What is also interesting to see is if you do this selection in a single pharmacophore way, that means only the single pharmacophore library, and you do it in a dual pharmacophore way, you find also you get sometimes very different fingerprints. So it's not only that you improve the binding, but you can also improve the fingerprints by doing that. We were also working, my interest is to work in different DNA, let's say, architectures and setups. One such idea was done in the past by a postdoc in our lab in Xiaoyu Lee, which is now a professor in Johnson University, and who was also the PhD student of Xiaoyu Lee. And we have shown that we displayed different building blocks like pins on a constant macrocyclic scaffold so that they projected towards the target protein like an antibody would with its complementarity determining regions. And essentially that was a test to see if that works. And you here see the fingerprint of the naive library, so unselected library, which is very homogeneous. And for different targets, which are not shown here, but you can look them up, you have very specific fingerprints, and we found even some compounds for very difficult proteins like TNF alpha, for which not too much small molecules are known to exist. These small molecules, or this, let's say, macrocyclic pins, also performed like antibodies in immunohistochemical stainings. For example, here our hit stained PSA in the same way than the commercial antibody did. So, but how do I see the future of those? The future lies, I think, in 4 areas, in my opinion: in improving the chemistry, in improving selection technology, in doing new scaffolding and encoding technologies, and then go into applications. So chemistry, I don't want to touch too much because that is a big field and more publications come out. In principle, that one wants to cover a larger chemical space, one wants to make more drug-like compact delts, wants to have new DNA-compatible reactions, and one wants to have high-yielding reactions, because the high yielding leads then to a larger purity and better performing library. In terms of selection, I've presented to you the normal approach like before, like an immobilized target, and then you do the incubation, so the panning procedure. You could go into a second round of panning, but in principle then you sequence and then you decode. That is the thing. But as you've heard before, new technologies might be useful. For example, photocross-linking, the assistance by a reversible or reversible anchor, and that are technologies which need to be done. In order to get it to work, as I've mentioned before, on cells, there were some publications claiming to get it to work on cells. However, I would say it's still a problem to be solved. And essentially to go into cells, there were also publications, but there I think it's even a farther step to go. So I believe that we are not yet at the stage where we can use cell selections in general for doing selections in cells. There are also approaches to work on beads, like in compartmentalized systems, where, for example, the compounds in such a micro-encapsulated system can be released from the DNA tag and then used directly for an enzymatic test. That was maybe done by Hegel. So scaffolding, as I said, we have single display and dual display libraries, as you've seen before. The single display libraries can be used for fragment-based screening approaches. But what I'm reaching at is essentially want to tackle somehow the space between the very small molecules, so the Pinsky-like compounds, and the biologicals like antibodies. I think there's a big space which needs to be addressed, and there were some kind of macrocyclic libraries produced in the past and are still produced. And I think in order to tackle this macrocyclic system, we have to be better in producing such macrocyclic libraries. And we will see if we will be successful in the future. As I mentioned before, dual display, we went from 1-to-1 to a 2-to-1 approaches, but we can also consider going into an N+N approach, and we can also consider to make this cyclic so that we can express, for example, a dual cyclic library, a linear on one side, branched library on the other side, cyclic on one side, linear on the other side, or a bicyclic library and cyclic library on the other side. So by doing things like that, we would like to expand the chemical diversity. So for summarizing, I would just like to say that that technology will be continued to develop, even if industry thinks it's already a finished story. But I think there is still something to do to obtain more specific and high affinity ligands. Also for difficult targets, which will be difficult to be addressed with very small molecules like protein complexes or PPIs. for targets on cells and also possibly inside cells. And I think that the combination of standard split and pool DEL setup, so in single-stranded format, as I mentioned before, combined with the dual display technology, enables features which are important, like photo cross-linking based selection strategies, dock and lock selection strategies, hit optimization strategies. It allows for placing two sets of pharmacophores on the target surface simultaneously, which can be an advantage, of course. And it allows for creating very large and diverse DELs for de novo selections. And if we get such small, that is actually also the aim of target 35, as far as I understand, if we get such high affinity small molecule ligands for proteins of interest, this will enable efficient tumor targeting therapies, for example, as small molecule drug conjugates or as bispecific drugs. And it will also help to look at the basic biology of the cell. And that, of course, will also nurture science. And with that, I would like to close, and I would like to thank my team at ETH. I would like to thank my former mentor, Dario Neri, and the PhilocChem team with whom we are still collaborating partially. And I would like to thank Alex for also collaborating with me. And I would like to thank my sponsors from ETH, the Swiss National Science Foundation, the Swiss Confederation, the Swiss Cancer League, and the European Union. And I would like to thank you for your attendance. Thank you.
Great, thank you so much, Jorg. Yeah, that presentation covered a lot of ground, and I think it provided a lot of the basics as well, which I'm sure the audience members really enjoyed having it all outlined, right? Because DEL has been done in a lot of different ways, and it can get pretty confusing as to what people actually mean when they say DEL. I'll ask a quick question. I've probably asked this before in the past, but I know that you've been in this field for a long time, and you might be one of the only people who have done both single and dual pharmacophore DELs. And so I get the sense from your presentation that you're leaning more towards the dual pharmacophore approach. But could you talk a little bit about the success rates between these two rather different types of DEL screening?
I think the conception to see it as either or is not correct anymore. I think the point is it used to be like that if we were talking about a one-to-one approach, which is a fragment binding approach. And there the success rate essentially is like in fragment-based drug discovery. So we have been able to fish out, let's say, interesting things. For example, a binder to AGP, which essentially is now used also kind of, which is consisting of two fragments which only simultaneously work together. So the individual fragments show no, let's say, individual binding, at least in a measurable way, and that is something that is of course desirable. And but as we are going away from that system of one-to-one and doing more dual display in a modular way, so that it can be combined with split and pool based libraries in a single stranded way, I think that has a big potential, especially for selection. I think what people do not usually take too much into consideration is it's not only done by making a library, it's also important to have the right selection methodology, and both are very important, especially if you want to tackle difficult targets like on cells and things like that. Xiaoyu Lee has explained that pretty well. And I think dual display is kind of a modality which enhances, let's say, the single pharmacophore library approaches.
Okay, so I have more questions about the single strand and how stable it is or what it's like to work with, but let's wait. We'll wait in the interest of time until the panel session, and we can bring that topic up. Okay, so last but not least, we have our third speaker for the morning or afternoon, depending upon which side of the globe you are on, Dr. Andreas Brunschweiger. He is at the University of Dortmund. That's my best German pronunciation, I would say. He is a group leader, and his group, amongst other things, provides expertise in the DEL world. he received his PhD at the University of Bonn, and he did a postdoc at ETH Zurich. His group, when it comes to DEL, and his title does say it all, but I'll add a little bit more. We in the world of DEL, there's chemistries that we can't use because the barcode ends up being destroyed. And Andreas noticed this very early on, and he's come up with some inventive techniques in order to alter the barcode or to protect it so that chemistries can be done and chemical space can be accessed, which the rest of us in the DEL field can't easily access or can't access whatsoever. And so the title of his talk is Chemically Stabilized DNA Barcodes for DNA Encoded Chemistry. And so Andreas, I'll hand it over to you with a reminder to our what appear to be very shy audience members that there's no way you don't have questions. So use your fingers and I'll attempt to type them in. Okay, Andreas, it's all yours.
Alex, thank you very much for this fantastic introduction and for giving me also the opportunity to talk about some of our research in DEL. And I would like to take up one point that York raised in his presentation on one of the last slides, where he also mentioned that chemistry is indeed one important facet of the library technology. I think I can even skip this introductory slide. Now we are obviously also very heavily engaged in designing and synthesizing these molecular chimera that consist of a phenotype, a chemically synthesized molecule, which is connected by a linker to a genotype, basically, that a DNA barcode that identifies the structure. And obviously the advantage is you can handle these chimeric structures as complex mixtures. You can assemble really gigantic, not large, but gigantic chemical space, and you can select it on target proteins in a single experiment, and you can read out by DNA sequence which compounds are enriched and are therefore likely binders and maybe even bioactive compounds. Here are a few chemotypes that have been published over the years, bioactive compounds, and you see basically they have been identified from really large library sizes. Now we are talking here about 10 to the 9, 10 to the 8 compounds. So definitely this technology gives you a handle on numerically large compound libraries. But most of the reactions, if you take a closer look at the molecules, they enrich modular sp2-rich structures. So the question is here, it applies to this technology, but it applies to all screening formats, all screening technologies, the chemical space coverage. And there we wondered if we can get access to scaffold structures, small, compact cyclic structures, mainly cyclic structures that display their substituents into space so that they are three-dimensional. So we are especially interested in these sp3-rich compounds. And just, by chance this week, the Nobel Prize was awarded to a scientist who was an MPI director not too far away from Dortmund, just I think 30 kilometers from here, Benjamin List, and he is one of the pioneers of organocatalysis, making such sp3-rich scaffolds in a stereoselective manner by certain catalysts. Also, David Macmillan got this Nobel Prize. So there's obviously value in making them. There's value in making them to access certain chemical space, but it seems also to be relevant then later when it comes to developing the compounds into something that can be used by patients. So the question is, how can we make them? And we did a screening of different DNA sequences that are, here we work a lot with a solid phase, I have to say, that are connected to a solid phase. We screened 4 different DNA sequences, 2 pyrimidine DNA sequences and 2 DNA sequences that contain also the purine bases, A and G, and we treated all of them the same with metal salts and with organic reagents. And you see, for instance, a treatment with TFA for some time, something that chemists would consider a mild treatment. 10% of TFA is a mild condition. You find this in the literature. This is really tough, and only the pyrimidine DNA could withstand this storm of acid, and the purine-containing DNAs were completely destroyed. Now, there was nothing left by DNA fragmentation. Yeah, we even observed DNA fragmentation, and the reason is obviously depurination. We also tested oxidizing reagents like the cerium salt, and there we could not even find the DNA afterwards. In the MALDI analysis, in ATGC DNA, we had to publish the remainders of an ATC sequence. But again, the pyrimidine DNA was completely fine, even with this tough reagent. And then scandium triflate was also quite tough on DNA when we increased just the temperature a bit. Then we also saw DNA fragmentation. So this tells you that certain catalysts that are applied under certain reaction conditions are destroying DNA. And obviously, these cannot then be used under these conditions for making the compounds that are encoded by the DNA barcode. And so this is really the limitations. No? You want to avoid undesired chemistry, and the undesired chemistry being the chemistry on the DNA barcode. This means we have to avoid depurination, fragmentation. We need to avoid deamination, which is a bit of a maverick because all kinds of conditions can do this, and it's difficult to detect until you sequence the DNA. So more difficult to detect by mass spectrometric methodology. And obviously also oxidizing reaction conditions are to be avoided because they form 8-oxopurines, which are absolutely mutagenic. Then the process, Jörg also mentioned this, the process that you operate with complex mixtures of starting materials requires you to really essentially use click chemistry type reactions that work really robustly. And another limitation is you build up the compound stepwise, so you need split pool compatible building blocks. And all of these three limitations have an effect on the chemotypes that you can make. We used a very drastic approach in our first version to get access to a larger scope of reaction, let's say reaction space. We truncated the DNA barcode down to an adapter oligonucleotide. We called this HEX-T, a hexathymidine adapter. We would then make a lot of chemistry on this adapter, and then afterwards encode the chemistry. We used this to make a peptidomimetic library by UGI chemistry. We teamed up with Alex Lömling, who's obviously an expert in these isonitrile chemistries. Made a proof of concept library, and with this we developed the process, the downstream process, the barcoding, the selection assay algorithm for readouts, and found a protein-protein interaction inhibitor of TYAP, protein-protein interaction. The advantage is you can do a lot of chemistry on such a adapter oligonucleotide, and you have good control because you would isolate the DNA tagged compounds, but it's not very efficient because you have to work with discrete HEX-T conjugates. So we wanted to develop a chemical barcode that has a stability, or a barcode that has the stability of the HEX-T. And there was already, I think that Jörg also cited this publication. There was an approach in the early time of encoded chemistry, where people used a combination of the pyrimidines, which we know are super stable, and 7-deaza-A for encoding certain peptides. And we thought to use this for our barcoding strategy. And at first we developed the barcoding strategy. We have the barcode directly on the linker to the molecule, and then we developed a barcoding strategy to encode these compound barcode conjugates with elongating them by other barcodes. So this works nicely, and we also showed in numerous experiments by PCR that you can amplify them, and also that they are readable, say that they do not introduce mutations. And we also found that this DNA barcode consisting of T, C, and 7-deaza-A is super stable to protic acids and also to many Lewis acids. really difficult to destroy the DNA, but it's possible. I will show you a bit later. So then in parallel, we developed a lot of chemistry for our strategy, which we call couple branch couple. So we would couple bifunctional starting materials to this barcode. We can optionally work on solid phase if it's necessary. So if you need to work under super dry conditions for some chemistry, then we would pool such conjugates and do the branching step, make a lot of primarily heterocyclic chemistry. We are also very fond of multi-component reactions in this step because this allows us to include a vector for the third reaction here and a diversity element. And then we get 3-cycle libraries with scaffold diversity in the middle step. And we have quite published a number of chemistries. I will just briefly mention them. Here we can do the UGI reaction, we can do UGI azide reactions. We have the Göbke reaction works fantastic in this, and also the Hollywood superstar a bit in my lab is this UGI Asa-Wittig reaction, because you get hold of this, the oxadiazoles, which are where we currently have a bit of a hype in medicinal chemistry on this scaffold, and we can combine these scaffolds then also with different exit vectors. We are quite especially working on this the Asa-Wittig reaction a lot. We also can do acid promoted reactions, obviously, because our DNA barcode just shrugs off the acid that you add. So the Biginelli reaction is in reach. We can do Provarov reactions. We have a handle now in access to rock chemistry, and also a very old friend of ours, the Pictet-Spengler reaction, works quite fantastic. And this is, for instance, an HPLC just of the crude, without any purification. So this works quite robustly. Also metal reactions work, but I have to say something here. So we could in principle do terbium promoted reactions to get pyrazoles. I'm a bit skeptical here because of the aromatic character, but also gold promoted reactions to pyrazolines and certain spirocycles work. And so definitely there is now a good scope to make scaffolds. The limitation, the Achilles heel of our barcode, as of any DNA barcode, it can be destroyed by strong nucleophiles. Hydrazine chemistry under certain conditions is not so advisable. You get an attack to the pyrimidines. So this is something, when you work with hydrazines in the field, you have to be careful. You have to be really careful. And as long as we work on solid phase, also alkaline conditions are impossible because you would cleave the DNA from the solid phase. But we are obviously also working now in solution with these DNA barcodes to get an even greater access to chemistry. What we can do with these shorter barcodes is we can use Bock chemistry as a purification handle. So here you see a starting material on the DNA. We do a Provarov reaction to build up a scaffold. We can even isolate the diastereomers that are forming. Here's one of the diastereomers. Bock on, it's purified. And then in solution, we would cleave off the Bock group. So this means if we have reactions that give us certain heterogeneity in the yields, we could still separate the product pool from the starting material pool. And now, if you have an access to quite a good reaction space, the question is, how can you find the reactions? No? How do we get access to novel reactions? Obviously, isonitrile chemistry is famous. Biginelli reactions, Pictet-Spengler reactions you learn in one of the early semesters. When you study chemistry, the question is really, how can we find reactions from databases that contain millions of reactions? And for this, we are currently developing a computer-assisted tool. We call it the Reaction Navigator. We are collaborating here with Reaxys. We got data from them. We would prepare the data from the database. We apply a filtering cascade to remove those reactions which are simply hopeless, which will never be compatible with DNA. And then we calculate the descriptors of reactions, and we cluster the reactions into different clusters. And then we look into the clusters for those reactions which are interesting to us. And now I'm coming back to the DNA encoded chemistry. So we synthesized in a proof of concept library. Here are the scientists who did it, Verena, Marco, and Mattea. Verena did the Ugi-Asa-Wittig reaction in order to make an oxadiazole library. Marco did a Povarov chemistry library, and Mattea synthesized the library based on the SNAP reaction. Here we collaborate with Jeffrey Bodie in order to get access to a greater scope of SNAP reagents. We also designed the barcodes for diversity and the building block selection. We also use cheminformatics in order to select the most diverse building blocks and to filter out, let's say, building blocks that have problems in medicinal chemistry afterwards. We also enrich the library with certain fragments. So in order to target protein classes, such as, putting in hinge binders for kinases. And then the question was, of course, a make or break question. Do the barcodes, which performed perfectly well in Sanger sequencing, also work well in more high throughput methodologies, such as the next generation sequencing, not to read out the barcodes? And so we synthesized the library, screened it, sequenced it also just by NGS. And we for this library, we developed or further developed an enrichment factor analysis tool so that we can directly find something like structure enrichment factor relationships. This is something that York also showed already. And Sylvia then built up a KNIME-based workflow, a data analysis tool, which allows us to recognize patterns in the compounds, to identify frequent hitters, which are also occurring in DEL, and then directly to dock the compounds that we find onto the protein surface for those proteins where it makes sense, which are rather the kinases, no? Those proteins which have a well-defined binding pocket. And what do we find? The chemically modified codes are readable in NGS. We see that certain barcodes are less abundant. No? We can hardly find them. But this is not due to problems in the code, but rather here are the building blocks that gave less abundant codes. And this is thiophane. Chemists will directly remark that this is a troublemaker in synthesis, and it is. And also such fluorines did not behave well in the synthesis of the scaffolds. These structures would obviously also make them problems in medicinal chemistry afterwards. So we are now also kicking out such building blocks. But this is a technical proof of concept. We screened then on MDM2, which is our model protein for development of methodology. And this is the one plot where we plot enrichment factors. So how often is the DNA barcode of a certain compound represented in relation before and after the selection procedure. And what we find is that certain, not really a clear scaffold preference, but when here you have the scaffolds plotted against the enrichment factor, but when you look at the cycle 3 building block, it was really clearly an indole that popped up. And those who know MDM2 know that this protein loves tryptophan, so the indole side chain of tryptophan. This is exactly what we found. So it was valid, and the process was also validated, and we could also here dock then with our workflow one of the compounds, an oxadiazole compound onto the surface of the protein. So this is then an in silico validation, if you want to call it like that. Not only on MDM2 we found something, but we also got kinases for screening. Here for one of those kinases, we could not find any hinge binder fragments. So we thought, well, that has not worked, the screen. But then we docked one of the oxadiazoles on the surface, and we got a fantastic docking score for the compound. And we are now also comparing DEL selection outcomes with the docking results by Pareto ranking. And for this kinase, indeed, we could see that the docking score and the enrichment factors went mostly in parallel. So this was also a fantastic finding. So conclusions are, we have now a DEL barcoding strategy based on chemically stabilized DNA and a couple-branch couple DEL design that gives us a good scaffold diversity. The workflows for screening and data analysis established, and we found compounds with plausible binding mode for diverse targets. The outlook is here. We are very active in encoded library design to make more reactions now accessible to DEL design. We are heavily investigating alternative reaction media, published a bit on micellar reactions. We are looking also now in screening, more in screening technology, data analysis, and also have our small MedChem projects to validate compounds and also to improve their properties. And with this, I would like to thank the team that is that has grown over the years. It's a fantastic team to work with. I thank in the faculty Daniel Rau and Herbert Waldmann, who are hosting my group, and we are collaborating heavily with chemists in order to make all kinds of, to investigate the potential of all kinds of chemistries for DEL design. And we are working, also collaborating heavily interdisciplinary and upstream and downstream of the DEL process. And you can buy also the DNA encoded library starting next year, obviously, as we have founded a company. And with this, I would like to thank you for the for staying with us here. And I would like to point your attention to a special issue on DELs, which I had the honor to guest edit, and where we have many fantastic collaborations also of the people you have just heard this morning or afternoon. Thank you very much.
Andreas, thank you. That was great. I have a quick question about the barcode that you're using at cycle 1, which is stable. And my understanding is you have a split size of 15 there.
Yeah, we had. Now we increased the split sizes, we are going into split sizes of up to 30. But then this is due to the fact that the building block availability, bifunctional building blocks, is lower, and that we also pool the reagents according to reactivity, so that we can guarantee something. But from then on, you can calculate it's straight way into really millions of compounds.
So what's your
recycle?
How hard would it be to get that first cycle split size up into the thousands or hundreds at least?
Hundreds. We would not do it. Hundreds. I would say it's now in reach to have split sizes of 40, and then we would do the reaction in parallel, you know.
I see, because they'd be pooled, and then you would use the cycle 2 tag to further.
Yeah, we are more fond of doing many reactions on them in parallel, and we restrict the cycle 1 currently to 30, maybe 40. You know? But then. And if we have, let's say, more building blocks for cycle 1, we would rather have 40 and 40 and 40, for instance.
Exactly. Yeah. Exactly. Oh, okay. So you could have a cycle 1 split size of really any size you wanted. You would just manage the tagging scheme. It would be encoded by a combination of cycle 1 and cycle 2 tags.
Definitely. Yeah. But I think the numbers here come. It's a bit like a triangle, no? Where the split sizes are increasing from cycle to cycle. At cycle 1, you would have few building blocks. This is also related to the commercial availability of bifunctional building blocks, which is, they are just much less than the monofunctional ones. And then you have also to kick out many that do not have the properties that we want to have. This is also always an issue, no? Building block properties. Then we would do many chemistries on these pools. And so this gets, so the current library scheme, for instance, is a 50 by 200 by 200 library, to get into the million range. And I think there's the focus of our research activities to increase the cycle 2 chemistry.
Okay. Yeah. So I don't know if we really have much more time for the panel, but I think we should at least try to address one panel question. Probably the first question that I mentioned at the very beginning, which was, is DEL a good choice for target 2035? And I thought maybe the best person to answer this would be the person who's coming most from the screening side of things, which would be York. So So York, you know about Target 2035, you know about DEL. Is this a match made in heaven or in hell?
Well, you know, Target 2035, I think I've been there at the brainstorming meeting in 2018, and it has developed already a little bit. And I think even there, it was already discussed, how should one get so many, let's say, specific high affinity ligands, and that as small molecules, because if possible should have the potential to be targeting intracellular targets. And the question is, how do you get there? You know, we know how inefficient, let's say, pharmaceutical industry in general was in the past, I don't know, to create at least large amounts of new chemical matter, chemical ligands, because our technologies is not perfect. The former technologies were not perfect either. So I think the best chance to develop small molecule chemical entities as ligands, in my opinion, comes from our approach of DNA encoded chemical libraries. There are also rational approaches. There is also, I don't know, fragment-based discovery. But also, I mean, virtual drug discovery is still not, let's say, I would say, a reliable source of getting chemical matter to.
Yeah. Is fragment-based drug discovery a good way to go for a target with no known ligands and no crystal structure? It seems
I don't know. I don't know. So it's also probably not so super high throughput. I mean, the advantage of DEL is that the throughput that is immense. You can create very large libraries, and you can screen them by, for example, affinity-based selections at once, which is something that otherwise does not exist. The only problem with our technology is that we have to have diverse, let's say, chemistry, diverse scaffolds, and we have to ensure good quality, because everybody can create nowadays a DEL, but the DEL is not a DEL. You know, you can buy DELs. There are suppliers popping up from the ground, which I don't know, make which have some DELs which they have just constructed, which were never tested by anything, you know, and I don't know. So there's a lot of voodoo also in our field. I think one has to concentrate on quality of both the chemical part of a DEL, but also of the encoding part of a DEL and of the selection of a DEL. You know, there are several aspects which altogether only make sense. And I don't see, let's say, at the moment, at least, a better technology towards getting a ligand against any protein than the technology.
Andreas, do you want to give us your take on the same question?
Yeah, I mean, DEL obviously competes with other technologies, and the pros and cons are very clear. Also, the other techniques, I would say, have a lead. I mean, most of the bioactive compounds that are also used as probes in chemical biology are coming from high throughput screens. So from the screening of discrete libraries, obviously their chemical space can be designed at will. Whatever you can synthesize, you can put into the library. But as York also mentioned, who has access to such libraries? It's mostly large organizations. That's of course one limitation. The second limitation is you need vast quantities of a target protein, the screening assay that works. So this is really tough. The fragment-based screening, I would say, is also a bit ahead of DEL, but as in case of high throughput screening, I would say it's because more people are engaged in this. That's it simply, no? More people are doing this. But then the limitation is also having quantity of proteins. So this can be really a limiter. And then when you have a fragment, what do you do with it? The effort to get from the fragment to a full compound can be enormous. No? And you see this with some of the compounds like, I think, Vemurafib, and then also this BCL inhibitor was made from fragment screens, and this was a major effort. So that DEL has truly advantages in terms of efficiency. It has still room to grow, in my opinion. This is something that York also mentioned. It's the chemistry, stupid, so to say, to make other scaffold really a diverse a diverse choice of scaffolds. Pfizer scientists called it actionable chemical space. So obviously you need to design into a certain chemical space. And there I see really the challenge. I mean, for us, it's also challenging, but it's possible to get some scaffold diversity in there, but it's a good scientific task, I would say, a challenging scientific task to design these libraries so that they are diverse and have certain properties. And from my opinion, I think also that for finding ligands, DEL is just a good choice. It's just a good choice. And it's a pity that we have so little activity compared to other hit-finding technologies in the DEL field. There are so few research groups in academia that are doing it. And also, I'm not so sure whether the pharma company has fully embraced the technology. So my expectation is with more learning about the DEL field, it will be even more productive than it was in the past.
So Suzanne, can I ask a couple more questions, or are we out of time?
I think so. Christina?
Yeah, go ahead.
Okay,
Yes.
great. So I would give you my take on that same question, except that I think both of you covered it very well, and we're all sort of on the same page. So I won't dive into that more. So York, is there a target class in the world of DEL that we can't run selections against, which is really hindering the field, or which, if we could target, would really open things up more for us?
I think DEL is not perfect. And to avoid any misunderstanding, I didn't want to criticize other technologies to create ligands. The only thing that I wanted to mention, if you want to have a high throughput method for thousands of proteins, then you need to have a quick method to do it. That is what I want to say. And I don't see a quicker method than DEL for
that is
achieving this.
No one in the field of fragment-based screening or HTS thinks they can run 100 proteins side by side.
Yeah.
No one makes that claim.
For individual targets, you can always get ligands from different technologies. This I didn't want to criticize. But as you said, you know, there are probably different target classes for which DELs at the moment are not optimal. And that is what I clearly see, you know, that we are looking at small molecules, and we are looking at kind of druggable targets as they used to be defined in the past. But if we have to look at the target 2035, we have different kind of proteins, we have protein-protein interactions. And I would not say that we are yet at this stage where we can really say, oh, all our attempts are successful. So we are far away from that. And that's why I say there is still optimization necessary.
Andreas, is there a target class that you find your collaborators or others come to you and talk about screening that you wish you could do, but you can't?
Yeah. I think I would start with what target classes would be preferred for DEL. And I think all the intracellular protein-protein interactions are clear candidates for DEL, where you really have to scan literally the surface of a protein to find a compound. Rational drug discovery has its troubles with these surfaces, so this would be my preferred one. But also more conventional or better explored target classes such kinases are still candidates, no? Because you want to look for alternative binding modes. The difficult targets are obviously cell membrane proteins, no? Because here you need a lot of know-how from the biochemists who maybe prepare such proteins for you on nanodiscs or maybe even on the cell surface. So this would be something where we really need more effort. And then tough targets are DNA binding proteins, obviously. Here probably you need very well-designed controls to find anything. But also RNA binding proteins might be more happy binding to your barcode than to the molecule here. Also, it's then probably a question of a well-designed control experiment to find really the compounds. But the focus I see in DEL is really the protein-protein interactions, which are insufficiently addressed. Not like ubiquitin ligases, for instance, a whole family of proteins, more than 500, I think, plus mutated proteins and so on and so forth. And you have little to no chemical space acting on those, and they are very fashionable targets nowadays, also because of the PROTAC. That is also something I would like to mention. This DEL blends seamlessly with all the molecular glue approaches that are out. Because you have you can just cut off the DNA barcode and can then put there anything basically, no? And this is something that really no other technology can provide. And it's inbuilt, no? It's an inbuilt character in DEL, and this is a fantastic. And there we are unique, and we should also focus on this a bit more, I think, in future.
So Suzanne actually has an excellent question. I'll let either one of you answer it. I'll let you choose. You know, if you're an academic lab, it's very hard to access a high throughput screening deck. What about a DEL screen? If you're an academic lab, how are you supposed to access DEL screening?
Well, DEL screen, I mean, there are already, let's say, attempts also by, let's say, stipulated by Ushi, where, for example, let's say to academic groups who would like to have, let's say, a ligand towards their protein, they can use an open access kind of library, and they can run this by themselves, and that is a cool system, I think. The disadvantage is maybe that, okay, some groups may not understand, because it's a complex thing, maybe a little bit more complex than one thinks at the beginning, to do the screening right, you know, so that you have meaningful results from your selections. And I think in the future there will be more and more publicly available libraries, and they will be screened by people who have targets, probably individually. The question is, yeah, how successful these libraries will perform, I cannot say. So, but I see that for academia, because it is not so expensive, it is a, let's say, very cool way to try to find your own ligand for your, let's say, protein for which you want to do basic research by yourself, and not to be waiting for somebody else to give it to you.
Mm-hmm. Andreas, what do you think?
Yeah, I think I would second then York's opinion. I mean, making the libraries needs a lot of expert know-how, but also handling them, no, for in the selection assay. And I would think that rather people who are experienced in this, or who come from an adjacent field like phage display or Selex, so who know how to handle proteins that are immobilized on the surface, might be persons who could work with such DNA encoded libraries in their own lab. Probably the better way forward would be that specialized labs not only make the libraries, but also do the selection with their workflows, no? With their protocols.
So we have one more question, and this is an oldie but a goodie, and it's along the lines of, are the hits you find with your DEL screening too big? So, you know.
Well, it depends on the target and what you want. You know, if you want to develop a drug for oral applicability, and then of course you might be right. In tendons, the libraries which are getting bigger, also getting bigger in terms of chemical, let's say, volume, so to speak, but if, as I mentioned before, if you want to target difficult targets like protein-protein interactions, I don't think you can go with a Lipinski-like library and you would expect to find a lot of hits from that. So I think it has to be tuned. So the libraries used will probably later on be tuned towards the application that you want, you know. And I agree, some libraries might lead to, let's say, molecules which are too big for Lipinski routes, and yeah, that's like that.
Andreas?
That's the reason. That's obviously the reason why we are taking the pains of developing taking the risk in manipulating the barcode, no? Because we see exactly this problem that if in order to get something that medicinal chemists would call actionable, you need certain properties in the compounds. And with the molecular weight, I think it depends really on the scaffold. When you have a nice behaved sp3 rich scaffold which has certain heteroatoms in it, so that confer polarity, then you can tolerate also a bit of a higher molecular weight. We see this also now in the clinic, that compounds with a higher molecular weight are there. And molecular weight is not the only property in a compound, no? that is important for clinical success or for cellular
specificity and high affinity is the key, and the oral availability or the appliance to the Lipinski rules, I think, is secondary. I think what we need for also for targeting in the future, that's what I've seen also in collaboration with Daraneri in tumor targeting for small molecule drug conjugates. You can target, for example, extracellular targets, and then it doesn't matter so much the Lipinski rules. It depends on the indication, of course. If you have cancer, you're willing to accept, let's say, also injections. But if you have a chronic disease, maybe that's less good. But that's what I want to say. I think specificity and affinity towards the target is key, no matter how big the molecule is.
Yeah.
But also the properties of the compounds are important. Otherwise, I mean, what a pharma company ideally wants to have is a compound that they can put into a pill and which can be dosed.
Yeah, that's what I say. You should not expect that always. You know.
But shouldn't we aim for the chemical space that we cover with our libraries to design into this? And then the question is, which chemistry, which combination of chemistry and building blocks gives us the coverage of that chemical space? And if we have to go outside certain parameters, again, which scaffolds could bridge this, no? That we have a certain size in the molecules, but these are still cell penetrant. There are examples from the literature. There are the fantastic works of Kielberg, who showed this for macrocycles, that certain designs can be taken up into cells and others not, or others are also then kicked out by the cells again. So I think really that chemistry is here the key to it. The proper combination of chemistry and building blocks. That is my opinion.
So I've seen a lot of HTS and DEL screens run side by side, and I think what I've seen coming from these exercises is that sometimes all the DEL hits are physically larger than the HTS hits. And sometimes the DEL hits are very competitive with the HTS hits. Sometimes you only find DEL hits and you don't find any HTS hits. And sometimes you only find HTS hits and you don't find any DEL hits. So that's the reality. And I think you can't expect any screening platform at the moment to always give you great hits against undruggable targets. So, the target is going to really define, more than anything else, is going to tell you what sort of hit you're going to find. So you want a really small, potent hit, don't screen an undruggable target, screen an easy target. But of course, then it's easy to find great hits, and then those hits aren't worth as much. So, that's the balance that the project team has to come up with. But I think as a general trend, though, DEL tends to find hits that are on the larger side compared to HTS, although that's not always true, but it tends to be true. And of course, as Andreas said, the only solution is chemistry. Yeah, so it'll be great to see more and more chemists try to tackle this problem.
Yeah, but I would also like to make here the point that the DEL field is branching out. No, I mean, one has a whole technology field and many barcoding strategies, many compound formats that are now there that have their strengths here and their weaknesses there. So one should not be too harsh and say that this is the way a DEL must be designed, but we should really accept this research field is now developing. It is branching out. and at some targets, one format might be the better, at another one, the other one might be better. So, yeah.
So we could go on forever, I have no doubt, but for Suzanne, it's extremely late. And so I would like to thank both of you, York and Andreas, for your great presentations and your willingness to answer all of these questions. And of course, to Xiaoyu, although he had to leave early due to a pressing engagement, I'd like to thank him as well for having given the first presentation. Again, my thanks to Suzanne. I know that behind the scenes, it takes effort to set these things up. There's been a lot of emails going around for many months. And to all of you who have signed in and listened, thanks a lot. And if you have any questions, I guess you can always try to email or this entire webinar will be posted somewhere so that it can be viewed later. So with that, Suzanne, is there anything left to say or anything we need to?
No, I just wanted to echo your thanks to the speakers and to the attendees, and of course, Christina for all her help. We hope to hear from you either by the, I think Christina had sent earlier on a little survey. If not, visit our website, target2035.net. And if you need to be connected to someone or have any more questions, we certainly can help you point you in the right direction. Okay, thanks everyone. Enjoy the rest of your day and afternoon.
Thanks, Susan.
Okay, take care. Bye.
Bye.